I will just try to make it simple. Biochemistry is a difficult subject, one of my most difficult subjects, pero I will just deal with what I know about the heart. So whatever is lacking, it will be discussed by Dr. Monsanto. So it's an aerobic organ. It is made of a muscle, and its activity is continuous. It's not intermittent. So by the time you are conceived from the womb of your mother until now, it's beating 60 to 100 beats in it. It does not stop. In fact, the heartbeat is a bit faster if you work, and it's a bit lower if you're asleep, but it does not stop. So it's not like the gastrointestinal system that will rest if you're not eating and will work faster after a meal. The heart continuously moves. It continues. And this is the reason why it needs a lot of energy. 40 to 50% of the cytoplasm is a rich mitochondrial matrix. And it is rich in mitochondria because the purpose is to produce ATP because it's continually working. The heart can metabolize fatty acids and bodies, glucose, pyruvate, and lactate. These processes that you will learn in lectures of Dr. Monsanto. But what is peculiar to the heart is that all the other muscles, all the other organs in our body uses glucose. Of the brain, it uses glucose as its main source. But the heart uses fatty acids. As far as I know, tissue and heart are the tissues in the body that fatty acid as its main source of energy. The liver can also send fatty acid, but not really producing it, but meaning other processes of producing energy. But it's the heart that's the organ that really uses fatty acid as a resting. Is my signal okay? Can I chat? Oh, Lord, have mercy. Dapat kasama pala yun sa dasal mo, no? So hanggang saan yung narinig? So, or continue, it is a lot of energy. It uses synthesis. So ito yung tanong natin, bakit fatty acid? Why not glucose? Like almost all organs in the body. So let's answer that question. During heavy work, there is an increase in consumption of glucose from limited glycogen stores. So resting, it uses fatty acid as a source of ATP or energy. But during activity, it can use glucose to form ATP. Okay. Now ATP is used 60 to 70. Wait lang, hindi ko makita yung picture kasi andyan yung face niyo. Okay. So ATP is used 60 to 70% for contraction, and 40% of ATP is used for ion pump. ATPs or sodium potassium ATPs pump. We have discussed this in physiology. So eto yung other ways to use ATP. It's not all used for contraction. Only 60 to 70% of ATP is used in contraction. The remaining 30% is being used by pumps. And this is the calcium ATP pump. It's the pump that moves calcium out. Also the sodium potassium ATP pump, which moves sodium and potassium back into the normal milieu inside the cell. So ATP can be produced by glycolysis, glucose oxidation, or fatty acid metabolism. But again, in the resting heart, ATP is produced through fatty acid metabolism, and it is different from other organs because almost body to produce I hope it's clear. So glycolysis, this is a form of, as an aerobic process, it will yield pyruvate to NADH to ATP molecules for every glucose that enters the glycolysis pathway via substrate phosphorylation. So we use glycolysis in the heart only when there is a much needed work. Let's say if the heart rate is more than 100 beats per minute. Is my signal okay? Can you hear me?
Medyo choppy pa, Dok.
Saan yung narinig? I'm so sorry. I-try ko kayang mag-use. Pangit din kasi yung data eh. Ayan, medyo okay na? Are we good?
Yan na, Dok, medyo okay. Pero kanina before glycolysis, medyo choppy na.
Before glycolysis?
Yes.
Ito?
Before ito. Yes,
Okay.
Dok, yan.
So I hope you can hear me. So, mindset na lang kayo. Hindi ko siya mababasa to go out from the slide. So ATP is used 60 to 70% for contraction, and I will show you later how ATP is used in contraction. The 40% of ATP is used in pumps, like calcium ATP pump, sodium, potassium ATP pump. You are ready because discuss this in physiology. ATP is used by glycolysis, glucose oxidation, and fatty acid metabolism. But again, in the heart, it is peculiar because compared to other organs, it uses fatty acid in its metabolism. It will only use glycolysis during activity. Okay? Before I move, did you hear that?
Yes.
Hello?
We can hear, Dok.
Or maybe I should turn off my video na lang, no? Para save ng energy. Ayan. Oh, pero hindi niyo ako, hindi man importante yung face ko, no? Anyway, glycolysis. So, it's an anaerobic process. It will yield to pyruvate to NADH to ATP molecules for every glucose that enters the glycolysis pathway via substrate phosphorylation. And glucose oxidation is another aerobic process. Pyruvate produced by glycolysis enters in the mitochondrial matrix as ApoA enters the tricarboxylic generate NADH and ADH2 molecules. The reducing equivalents enter the electron ATP molecules via. So 38, it's an aerobic process. So fatty acid metabolism is also an aerobic process, but it uses a little bit lower oxygen as compared to the glucose oxidation. So fatty acid metabolism within the mitochondrial matrix, fatty acid undergoes several rounds of beta oxidation to yield a large molecule. So let's try to compare glucose versus fatty acid metabolism. So ATP, there are 38 ATP in glucose oxidation, but in beta oxidation or fatty acid metabolism, it produces 44. So mas maraming ATP, and this is only the reason why the myocardium uses this kind of metabolism, because it will produce more ATP. However, if you look at more as to energy efficient, hindi siya energy efficient, but it is more oxygen efficient because it will only take up 5.5 ATP for every oxygen molecule consumed. And so fatty acid synthesis produces more ATP and consumes less oxygen. And this is the reason why genetically our myocardium uses fatty acid synthesis as a source of energy because it produces more ATP and it consumes less oxygen, while the rest of the organs, especially the brain, uses glucose, okay, as a source of energy. Did you hear that? Was this okay?
Yes.
Doc.
We answered that question, why the heart uses fatty acid synthesis as not glucose synthesis as compared to other organs. Kasi halos lahat ng organs ng katawan, it uses glucose as a source of energy. Happy. Okay. But did you understand why the heart uses fatty acid synthesis rather than glucose oxidation? Were you able to get that?
Yes, Doc.
Yes. Yes po, Doc.
So, lang, the myocardium or the heart uses fatty acids because it uses more ATP. This process produces more ATP while consuming less oxygen. Okay? I hope that is clear. Okay. So, in myocardial ischemia, there is less oxygen. So, the fatty acid synthesis will be helpful kasi konti lang naman yung oxygen na nakoconsume niya. But there will be a decrease in ATP synthesis and increase in lactic acid. And it is this increase in lactic acid that causes angina. So it would have been beneficial if we can use glucose oxidation during ischemia. However, you cannot just genetically shift from fatty metabolism to glucose oxidation. You cannot shift in one day or in a few hours after myocardial infarction. And this is the reason why you need medicines that could inhibit and stimulate glucose oxidation. So we have medicines like aminoacidine that the myocardium use glucose oxidation to form ATP. Okay, so yun lang. So if you look at the internal structure of the heart, you have the thin filament, which is the actin. So my name is actin, and which is myosin. You need the myosin head to attach to actin so that the bone so that shorten. Okay. So far? Hello? Bakit parang nang nag-lecture ako last time, okay naman, no? Tapos ngayon, choppy na. Okay. So, hopefully, we can finish this lecture. So, somebody should tell me if it's nakukuha niyo or not. So, in order for the myosin and the actin to move in a contraction, it needs calcium. It needs calcium. So, I have discussed this. So, calcium, cellular space, 20% will enter through L voltage gate channel towards the cytoplasm, and then that 20% of calcium will attach to the ryanodine receptors, and it will cause the 80% calcium to root down the cytoplasmic reticulum. So that you have a lot of calcium that will go into the actin and myosin to act in the contraction. Okay, so there's also a process of removing calcium so that you will go into diastole, because during systole, that's where calcium is released. And during the diastole, the calcium will now be brought back to where it belongs. 80% will go back to the sarcoplasmic reticulum through the, and the remaining 20%, some it will go out through the sodium calcium exchanger or to the calcium ATP pump, or a little bit of 1% will go into the mitochondria via the uniport, and this is to be used in production of ATP, which I will discuss in a while. Narinig niyo? Hello? signal so far? So, na-discuss natin to before in physiology, the importance of calcium in contraction. The internet is unstable, Lord Jesus. Okay. So, the action potential is conducted along the surface sarcolemma and sarcolemma that extends into the T-tubules, and calcium at sites of junctional sarcoplasmic reticulum cleft trigger local calcium release, and the calcium diffuses throughout the cytosol to activate myofilament contraction. So, una muna, 20% of calcium will enter from the sarcolemma, meaning outside from the cell into the cell. And this 20% of calcium will now attach to the sarcoplasmic reticulum through the ryanodine receptors, and it will cause release of calcium from the sarcoplasmic reticulum into the cytosol to activate the myofilament contraction. I hope that is clear. So the calcium quickly declines at each beat because of calcium uptake via the sarcoplasmic reticulum pump, or what we call as SERCA, and extrusion by sarcolemmal sodium calcium exchanger and calcium ATP pump. So, and also into the mitochondrial calcium uniport. And then you will go into diastole. Okay? Naintindihan so far? Hello? All right, all right. So the myofibrils are bundles of contractile proteins that are organized into a regular sarcomeric array bounded longitudinally by V-lines that are immediately adjacent to tubules that run in parallel. So in diastole, the thin filaments, meaning the actin, create a cage around the thick filament, which is the myosin, that have cross bridges, or the myosin head, that extend toward the thin filament. Myosin molecule tails all face the center of the sarcomer, creating a zone around the M line devoid of myosin head. During systole, the myosin cross bridges pull the thin filament cage toward the M line, thus shortening the sarcomer in length. Okay, so this is just trying to show you that the 25% of calcium is coming from the external surface or extracellular space through this voltage L-gated channel, and then it will stimulate, it will attach to ryanodine receptors, and then will release 75% of calcium into the cytoplasm, and this calcium will now be needed for the contraction of calcium. And again, after that's systole, then during diastole, 75% of calcium will go back to the sarcoplasmic reticulum through the SERCA, and 20% will go out through the sodium calcium exchanger into the extracellular space, and then 1% into the calcium ATP pump, and 1% will go to the mitochondria via the uniport. Okay? So calcium entry via the voltage activated L-type calcium channels triggers the release of more calcium from the sarcoplasmic reticulum, and a tiny amount of calcium that enters via the sodium calcium exchange early in the action potential. The removal of calcium ions from the cytosol is mainly via the sarcoplasmic reticulum ATP pump, or SERCA, that's 75%, and sodium calcium exchanger, that's 24%, and tiny amount is transported to the mitochondria via the uniport and the sarcolemmal calcium ATP pump, another 1%. All right. So the sodium potassium ATP pump extrudes the sodium ions that enter during the sodium current and the sodium calcium exchange action. Okay? Pare-parehas na atang information dito, no? So dito na lang tayo. I have been talking about 1% of calcium that enters into the mitochondria. And this is an inner process that calcium enters via a uniport into the inner mitochondrial membrane. And this calcium that enters into the inner mitochondrial membrane will be extruded outside into the cytosol via the sodium calcium exchanger, In this process, one calcium will go out, but 3 sodium will enter. And we don't want sodium to be inside the inner mitochondrial matrix. So sodium has to be extruded out. And this is done through the sodium H plus exchanger. So in this process, sodium will go out, but protons will go inside. This process is not energy coefficient because we want protons to enter into the mitochondrium through the electron transport chain. Okay. However, this H plus will now be extruded out into the cytosol through the cytochrome processes. Okay. Ang tanong, bakit na ba pumasok si calcium when there are a lot of processes of entering sodium and extruding out sodium, and then H plus enters again and H plus is extruded out, all for one purpose, and that is to enter calcium, 1% of calcium. So because calcium is needed to activate an increase in dehydrogenases, there will be an increase in NADH, which will be used by the F0, F1 synthase and eventually produce ATP. Okay, that 1% of calcium that enters into the inner mitochondrial membrane is actually needed to produce ATP. And the discussion about electron transport chain, the F0, F1 synthase will be lectured by Dr. Monsanto. Did you hear me? Was it clear?
Yes.
Hello? Okay. So you just tell me if hindi nyo naiintindihan kasi I always ask questions and then try to answer a question. So my first question was, bakit the myocardium uses fatty acid synthesis in production of ATP? And I've answered that. That's because fatty acid synthesis produces more ATP and uses less oxygen. And this time, I was also wondering, bakit you only need 1% of calcium into the mitochondria? But in that process, there are a lot of mechanisms that happens afterward. Because one calcium enters into the inner mitochondrial membrane via the uniport, but that calcium has to be extruded out. So in that process, you need the sodium to enter because the calcium can only be extruded out through the sodium calcium exchanger. And in that process, sodium enters inside the mitochondria. And mitochondria will be extruded out through the sodium proton exchanger. And in that process, there are protons that goes inside and it will be extruded into the COM process. So the reason why we need calcium to enter into the inner mitochondrial membrane is because it increases dehydrogenases, it increases NADH, which will be used in the production of ATP. Okay? Okay. So, ganun lang siya. All right. So, we have been talking about calcium that enters into the cytoplasm for contraction. So, you have the myosin head, and then you have the actin. So, the actin is the thin filament, the myosin is the thick filament. So, the myosin head has to
Choppy po, Doc.
Oh, no. Let us pray.
Choppy.
Ayan, okay na. Back? Okay, okay. Kung ganito siguro na makakatipid siguro no ng energy. Ayan, okay na? Can you hear me?
Yes, Doc.
Okay. So the myosin head has to attach to the actin in order for the myosin to move or pull the actin towards the center and shorten the sarcomere. That's during systole. So the myosin head has an attachment site in the actin. Okay. So that active site is covered by this troponin complex. So you have troponin C, which is the binding calcium. This troponin C is troponin I, and this troponin I is attached to troponin T. Okay, so the troponin I stabilizes troponin T. So when troponin I troponin T, it will cover this tropomyosin. The troponin kasi is active, is the binding site of the myosin. Oh, Lord have mercy. Okay. Wag na nga lang mag-share. Ikaw muna mag-share. Ayan, you can hear me? Can you see me?
Yes,
Doc.
Yes,
No.
Doc. Opo, Doc. We can see you.
Yes po.
So we have troponin head and we have the actin. So you need the myosin head, the myosin head and the actin. So the myosin head has to attach to the actin, okay? So that the myosin head can pull the actin towards the center and shorten the sarcomer. And this is during contraction, during systole. So the myosin head has an attachment site on the actin, and that is the active site is the tropomyosin. But that active site is covered by troponin complex. Okay? So this troponin complex is made up of troponin C. C because that's where the calcium will attach. Okay. Then the troponin C is attached to troponin I. And troponin I is attached to troponin P. So yung function ng troponin I is to stabilize the position of troponin T. So if troponin T is positioned, stabilized, or it's stable in the tropomyosin, the tropomyosin is covered by troponin T. Is it clear? So if tropomyosin is covered, that active site, meaning the myosin head cannot attach to the actin and no contraction will happen. So in order for contraction to occur, yung calcium na pinag-discuss natin kanina, na lumabas from the sarcoplasmic reticulum, 70%, tama, 75%, and the 25% coming from the extracellular space, 100% lahat ng calcium. So that calcium will go to troponin C. It will bind to troponin C. So once troponin C, once calcium binds to troponin C, it will now stimulate troponin I. Parang there will be conformational change in the troponin I. The troponin D is
Copy, Doc.
I and my
Copy again.
Lord of mercy. Ayan, okay na? Ha? Kung hindi ako nakikita, parang gusto ko kasi nakikita ko kayo. My Lord, dapat ba ako nasa ospital? Ano ba? Wait lang. Wala nang ibang nakakonect. Ako Hello, am I back?
Yes po, Doc.
Yes, Doc.
Ay, nakakahiya. Diyos ko, Lord. Nakakahiya. Okay. Let's try to just move on. Okay, so you have the troponin complex. Okay, it has troponin C. This is where calcium will bind to it. And this troponin C is attached to troponin I. This troponin I stabilizes troponin T, and troponin T is the troponin which is active site for binding of the myosin head. Okay. Any so far? OMG. I'm sorry. Hello?
Hey, Doc.
Pag, pag nag-i-start ng magsalita, no, nawawala na. Okay. Anyway, so lesson learned. Hindi na talaga ako aasa sa Wi-Fi, no? Tridor yung Wi-Fi ng Globe ngayon. Nagsisisi tuloy ako. Dapat nasa hospital ako. Mas malakas doon. Mas powerful, no? Okay. Hindi siguro bet ng Wi-Fi yung dinidiscuss ko na troponin C. Troponin I, troponin C. But anyway, in a small concept, parang calcium will just bind to troponin C, and troponin C will stimulate the troponin I to stimulate the troponin C to move away from the binding site, and the calcium will now the troponin head will now bind to the actin. Okay. Hi. Wait lang. Kailangan kasi natin mag-ano eh. So, okay. So we have discussed this in physiology. All right. So, ang next event would be to produce this power stroke. Okay. So the first stage is the rigor state. That's where the myosin head is attached to the actin. And this is rigor state because the myosin head will not move away from the actin without the presence of an ATP. So when another ATP arrives into the myosin head, the actin will try to move away from the myosin head so that the myosin head can bend over. Okay. So when when calcium arrives, that's when the active binding site in the actin will be removed, and then the myosin head can now attach to the actin. And so because you have now the ATP, it can now create the power stroke wherein the myosin head will bend over and pull the actin towards the center. Okay? So after that, the myosin head is attached to the actin. Without the presence of another ATP, the actin will not move away. So ito yung tinatawag na rigor state. Did you hear me? Was it clear? Hello?
Yes, Doc.
Okay. So have you heard about rigor mortis? Have you heard about it?
Yes,
Doc.
Yes, Doc.
Yes, Doc.
So in rigor mortis, naninig it patay, and that is the reason why, because it is in a rigor state, meaning the myosin is attached to the actin. So myocardial ischemia, for example, there will be loss of ATP production or myocardial infarction or namatay ang pasyente. So there will be loss of ATP production. And again, without the presence of ATP, the myosin head is attached to the actin. And that's the reason why patay diri na si kahumok. Okay, that yung tinatawag na rigor mortis, because the actin is in a binding, is bounded or yes, it's bounded or attached to the myosin. The myosin head is attached to the actin at all times. Hindi sila nag-separate. So it's in a weak binding site. Yun yung rigor state. Okay, so without the presence of another ATP or a new ATP, the myosin will not move away from the actin. So pag hindi kasi siya mag-move, it cannot bend backwards so that when the actin will attach to it again, it can bend or move forward to pull the actin towards the center. So yun lang yung concept ng power stroke and yung presence ng calcium and the presence of ATP in the contraction. Okay? Yun na yun. That's just the statement. Kasi mahilig kasi akong mag-explain ng picture. Every time nag-explain ako ng picture, pag walang statement, yung mga students din na nag-aaral, tapos kaya nilalagay ko na lang yung mga statements para pag-share yung slides, kawawa naman, may nababasa na sila. So, yung in-explain ko na yun lang yun yung nasa sa slides. So, we have been talking about troponin C, troponin I, troponin C. This is very useful clinically because if there is myocardial damage, you release troponin I, troponin C into the bloodstream. At pagkuha yung pasyente ng dugo in the cubital vein, antecubital area, parang ganon. So, pag mataas yung ng troponin I or troponin C, then we know there is myocardial damage because this troponin C and troponin I are high in the myocardium. Okay, so we can measure that and assess for presence of myocardial injury or myocyte injury. So this is helpful in the diagnosis of myocardial infarction because troponin I will rise within an hour after myocardial infarction and it goes down after 7 days. So whereas myoglobin or CKMB, which is also inside the muscle of the myocardium, it immediately rises about 30 minutes. But in less than 1 hour, it declines immediately. So we can use CKMB in the diagnosis of myocardial infarction for its acuteness, as well as troponin. So nagagamit lang natin, ngayon, what we use is just troponin I or the diagnosis of MI because mas mahaba yung presence niya in the blood. It can last up to 7 days. Now, I can only use troponin M, CKMB. For example, I had admitted the patient for 2 weeks and on the troponin I on admission was elevated and the CKMB was only slightly elevated. But then after 2 weeks, the patient had another chest pain while on admission. So when I request, not after 2 weeks, the patient had another episode of chest pain. So the question is this chest pain another new MI? Ang gagawin natin is we request for a CKMB because if the CKMB rises again, then we know it's an acute MI. Kasi if you request for another troponin, mataas pa yung level ng troponin at 4th day. Did you understand my example?
Yes, Doc.
Kailangan bang ulitin?
Yes po, Doc.
So,
Yes, Doc.
kasi last 4 days. So if you request troponin I within 7 days, talagang mataas siya and you cannot diagnose if there is an acute event. Unlike CKMB, it's only less than an hour. So if the patient had another chest pain on 4th day of hospitalization, if you request for a CKMB and it has increased farther, then you can understand that the chest pain of the patient is a new or an acute event of myocardial infarction. So yun lang, yung importance ng CKMB at saka yung troponin. Okay? Short lang. So troponin I rises within 2 to 4 hours and it normalizes within 7 days, while CKMB rises within 4 to 8 hours, but it will normalize within 3 days. So we can use CKMB in patients for diagnosis of acute MI. But if we compare the two, troponin I or troponin T versus CKMB, mas specific is troponin I and troponin T because there are other muscles that can also release CKMB, not just the myocardium. Okay. Tapos na. Yun lang. It's a very short topic lang. So just to recap, kakaiba lang yung myocardium because it uses fatty acid synthesis in the production of ATP, because fatty acid synthesis produces more ATP and it is oxygen efficient. It uses up less oxygen. However, in myocardial infarction, there are you want to use glucose as a source of energy somehow because mas maraming glucose. Okay, so it is energy kasi it's glucose kasi is more energy efficient. However, you cannot shift your genetic composition from fatty acid synthesis to glucose oxidation in a matter of few minutes. So you need medicines to help you augment glucose oxidation in the myocardium to produce ATP. Okay, now calcium is needed for contraction in the actin and myosin filament interaction, and you need that for the binding of calcium into the troponin C. And in this process, the troponin complex will move away from the active site, and the myosin head can now attach to the actin. And with the presence of ATP, you can create a stronger power stroke. And without the presence of ATP, the actin and the myosin is bound to each other, and this is what we call as rigor mortis. Parang it's in an active, it's in a weak bound. Parang they are always bound together, and without the presence of another ATP, they will not be released and be able to produce a stronger power. And this troponin I, T that we have been discussing in the troponin complex is helpful clinically in patients with myocardial infarction because it's highly specific. If you see an elevation of troponin I or troponin T, you know that there is myocardial ischemia. CKMB is also used. However, this is not as specific as troponin I or troponin T because there are other muscles that release CKMB. However, we can use CKMB in the diagnosis of acuteness of the presence of myocardial infarction. So these are just the concepts that I want you to understand. Questions? So yung ibang portion of mechanic or yung biochemical reactions in the myocardium, I've been trying to discuss it during my discussions in physiology. So na-augment naman. So kung anong kulang, dito siya. Okay, so we will be still discussing about myocardial infarction. So I think there are still more information that we can get from there. So yun lang. Sorry for the bad signal. Minsan naman pag nagle-lecture ako, okay naman yung signal, pero minsan pangit. Are there any questions? Bakit nang patapos na ako, no? Parang okay yung signal. Kanina pangit.
Doc, question, Doc.
So nawara niya signal kahit question niya.
Hello, Doc.
Yes, yes.
Doc, di ba during during systole, calcium is released from the mitochondrial matrix. I just want, kasi kanina, Doc, di ba, there are percentages of calcium released during systole. And I just wanna, Doc, I just want to, kasi medyo nag-choppy kanina when you were explaining it. So, just yung mga percentages lang. Yun lang. I just want to, know.
25% of calcium is coming from the extracellular space. Nag-choppy siya. Hello?
Okay,
Yes,
okay,
Doc. I can still hear you, Doc.
Percent of calcium is coming from the extracellular space. It's outside of the cell. And the purpose of that calcium is to stimulate the sarcoplasmic reticulum to release a lot of calcium. Kasi the sarcoplasmic reticulum is an organelle that stores a lot of calcium. Kasi yung 25% na calcium coming from the extracellular space is not enough for the contraction coping. You need a lot of calcium para lahat ng troponin C ma-activate mo, and then para mag-move away, and then yung troponin complex so that the actin and myosin can now interact and cause the power stroke. So that 75% of calcium is produced to you by the sarcoplasmic reticulum. Luckily, the sarcoplasmic reticulum, yung parang key niya to release calcium is the presence of calcium. It's the calcium that will bind to ryanodine receptors. And once there is presence of calcium in the ryanodine receptors, calcium will be released from the sarcoplasmic reticulum. And this calcium will now go into the cytoplasm and cause contraction.
Thank you, Doc.
75% from the sarcoplasm from the cellular space. What is my reference of book? There is lesser discussion of myocardial, yung biochemistry kasi ng heart is hindi siya well discussed in our book ng biochemistry. So my source is our Bible in cardiology. So is my source. Somebody's asking. So pakirepeat lang po yung trop-I at CKMB. So meron tayong troponin C, di ba? Ano, may troponin I, may troponin T. Okay? So, so these are biomarkers for diagnosis of myocardial infarction because these are present inside the myocardium. And when there is myocardial injury, troponin I and troponin C can release into the body. So, you can diagnose if the troponin I or troponin C is elevated, you can diagnose for the presence of myocardial infarction. However, there is that extent, troponin I can last up to 7 days in the body, while CKMB can only last for less than 3 days. So we use troponin I better than CKMB because the specificity is better, is higher in troponin I as compared to CKMB. For example, the patient has sepsis, or the patient has tumor, and CKMB can rise, but not troponin I. So hindi ganun ka sensitive din si CKMB, and that's why we use troponin I. We can only use CKMB in diagnosing the acuteness. Kasi since CKMB decreases within 3 days. So, for example, the patient had chest pain at 7th day. If you request troponin I, it's an acute event. So you need to request for a CKMB because if the CKMB is high, then you know that there is an acute event. Parang nag-MI siya on day 1, nag-MI na naman siya on day 7. Yun lang yung beauty of troponin I. Hello? Okay ba? Did I answer, Dale? Okay. Katrina, you were asking the process where calcium attaches to troponin C. So We have been discussing 75% of calcium released from sarcoplasmic reticulum. If this calcium is released into the cytoplasm, it will now attach to the troponin C. It will just attach to the troponin C because the binding site of calcium in the actin is the troponin C. So once troponin C, once calcium attaches to troponin C, there will be conformational change of troponin I and troponin C. It will move away. The troponin C will move away from the tropomyosin. Tropomyosin is the active site for the myosin head. So if the binding site of the actin is open, myosin can now attach to the actin and can now go into the process of pulling the actin towards the center, shortening the sarcomere. That's during systole. Answer your question, Katrina? Okay, you can ask anytime. Alam niyo naman ako, open for questions. Hindi kasi ako nag-discuss nito ng troponin I, troponin C. That's why I needed to discuss this. Hindi ako yung nag-discuss niyan. That's why I need to just augment this during this discussion. Any more questions? Hello? Are we good? Okay na tayo? All right. Thank you very much. Good afternoon. I am so sorry. I really cannot explain bakit rinatry-door ako ng Wi-Fi.
Thank you, Doc.
Nga si Globe. Thank you.
Thank you, Doc.
Tomorrow we will.
Thank you po.
Thank you, Doc.
Thank you, Doc.
Thank you, Doc.
Thank you, Doc.